welcome you all. I'm your host for the session today. My name is Neelam Mehra, and today I'll be sharing with you top 10 tricks to revise Unit 3 faster. The whole purpose of these video series, where we are giving you tips to revise unit-wise syllabus of CSIR NET, is majorly to help students to utilize these few days for their preparation for the exam. With this agenda in mind, let's begin with the session today. So there will be free sample papers that you can download from the link given in the description below. As we know that the Unit 3 majorly deals with the fundamental processes of life itself. So this unit is one such unit where you can fetch very high score as it is whole conceptual based. So to build your concepts and get a good hold in this subject, you need to go through the standard books written by the foreign authors, such as Molecular Biology of Gene by James D. Watson, Molecular Biology of the Cell by B. Albert, Leringer, Principles of Biochemistry by David L. Nelson and Michael M. Cox, Cell and Molecular Biology, Concepts and Experiments by Gerald Karp, Molecular Cell Biology by Harvey Lodish. So let me tell you why you shouldn't skip this unit. This unit is very essential since it includes all the fundamental processes of life itself. Students find this unit very simple as it is all conceptual based and they find it interesting to go through the subject and read it. This subject is very scoring if you have built your concepts very well. So leaving this unit won't be feasible. On an average, 10 to 12 questions are being asked every year in CSIR NET exam, both in Part B and Part C. Concepts are being merged with Unit 1, that is Biochemistry, Unit 2, that is Cellular Organization, Unit 4, Cell Communication and Cell Signaling, Unit 8, that is Inheritance Biology, and Unit 13, that is Methods in Biology. So since this unit, the concepts in this unit is linked to so many units in the CSIR NET syllabus, leaving this unit is not recommendable. Then coming up to the major topics that you need to learn and revise from this subject. First comes the prokaryotic and eukaryotic DNA replication, where you need to study all the three models that are involved in DNA replication. That is conservative, semi-conservative, and dispersive. The experiment that supported the semi-conservative nature of DNA replication given by Meselson Stahl. Prokaryotic and eukaryotic origin of replication, replication fork, fidelity of replication, and extrachromosomal replicons are important. DNA polymerase being the core enzyme used in replication machinery. The types and differences. You need to also know about the other enzymes involved in replication mechanism, such as helicases, gyrases, DNA ligases, topoisomerases, etc. And telomerase is also an important topic that you need to cover. Second subunit comes is DNA repair pathway, where you will be coming up with the concepts of types of mutations, the sources of mutations, various types of mutagens that are involved in damaging the DNA, and what are the different types of DNA repair pathways that gets activated to check on these damages, these mutations. The enzymes that are involved during these DNA damage repair mechanism. What is the source repair mechanism and translation repair mechanism? Then comes RNA transcription. In this, you need to study about the types of RNA in the cell. Second comes the RNA polymerases and their structural components involved both in eukaryotes and prokaryotes. Third is the transcription factors that are involved. What is their sequences of events and what are their different roles? Then comes the role of promoters, enhancers, activators, repressors, and mediators. Another important subunit is about post-transcriptional processing, where you need to remember about the events and the components that are involved during the processes of capping, polyadenylation, RNA splicing, and RNA editing. Then comes about genetic code, where you must remember about the start codons, what are the stop codons. Another important subunit is translational inhibitors. What is the mode of action of these different types of antibiotics? How and where they exactly work or act upon during translational event? This you need to be thorough with. Then comes the OPERON model, where you need to study about the lac operon and trip operon majorly. In lac operon, what are the different types of genes involved in lac operon? What is the catabolite repression? In trip operon, what is the attenuation regulation in trip operon is important, and the mutation effects on operon. Then comes eukaryotic gene regulation, where you need to be thorough with the chromatin structure, role of methylation and acetylation in gene expression. The gene regulation during development can be merged with Unit 5 when asked in exam. Unit 5 is basically developmental biology, so you can study both of these concepts together. Yes. So then comes the semi-conservative model of DNA replication given by Meselson and Stahl Experiment. So here both numerical and statement-based questions, including the hypothetical ones, are often being asked in Part C. So you can attempt the previous year question papers which deals with these type of questions on semi-conservative model of DNA replication given by Meselson and Stahl Experiment. These kind of questions, attempting these kind of questions, you will have an idea like how to solve the concepts and how to build the concepts. Then comes, you need to cover all the three models of the DNA replication, that is conservative, semi-conservative and dispersive. The results of the semi-chloride density gradient method as an evidence of semi-conservative model is also important to study, because graphical based questions or experimental based question comes from this section. Yes, the types of polymerases. So if you want to memorize the types of polymerases that are involved in DNA replication machinery, you can make use of comparison tables such as I've given here, where I am comparing DNA polymerase 1 with DNA polymerase 3. Here you can remember the names and the function of different DNA polymerases, both in prokaryotes and eukaryotes respectively. Fidelity of DNA polymerase is also very important. You cannot skip this part. Yes. Then comes the enzymes at replisome. What you can do here is you can make and keep a colorful diagram of replication fork depicting the bidirectional mode of replication. Both numerical and statement-based questions are being asked from bidirectional replication model and Okazaki fragments. You need to enlist all the enzymes working in replication fork during replication mechanism. Majorly, the match the following questions are asked on the basis of the functions of different enzymes during replication. So let me give you an example here. What kind of question can be asked, the numerical based question. So here it is given that there is a diploid organism which is having a genome size of about 4.5 into 10 to the power 8 base pairs. And the DNA, the whole genome, whole DNA is being replicated in 3 minutes. Assuming that all the replication forks move at the rate of 10 to the power 4 base pair per minute, how many replicons or replication units are present in the organism genome? So it's a very simple question which is being asked in the form of numerical that you need to solve. So what you can do is you can pause the video, think for yourself how can you solve this question, and write your answers in the comments given below. Next comes the prokaryotic and eukaryotic replication mechanism. So here what you can do is all the replication events, you should learn them through flowcharts or simple diagrams. It would be very helpful for you. The sequences of replication events is most of the times being combined with cell cycle, that is from unit 2, and graphical based questions are being asked in part C from this section. So you can, for this also you can take the previous year question paper and see, look for yourself what kind of questions come here so that you can get a clear-cut idea, and it would be simpler for you to solve these kind of questions later through practice. Then comes the DNA repair pathways. So here what you need to do is you need to remember all the 3 main categories of DNA repair pathways. First is the direct reversal. Like what are the kind of events that are going on here and what are the enzymes that are involved. Both of these things you need to remember respectively for each of the different types of DNA repair pathways. First is the direct reversal, in which 2 types come, photoreactivation and methyl group removal. Second is the single-stranded damage, where base excision repair is involved and nucleotide excision repair is involved and mismatch repair is involved. Thirdly comes the double-stranded breaks, where non-homologous end joining, you need to remember what is the events going on and what are the enzymes involved. Homology-directed repair. Third is the microhomology mediated end joining. Then comes DNA damage and DNA repair pathways. For this, you can use the comparison tables for differentiating between different types of repair mechanisms, such as what is the difference between base excision repair and nucleotide excision repair. I have given here as an example in the form of comparison tables. You can also do the same for yourself. And you can also make comparison tables for translation polymerases and SOS repair mechanism. Don't miss the types of mutations and mutagenic events that causes these kind of DNA damages, what is their effect on the genetic construction, and which kind of DNA repair mechanism gets activated due to these mutations. All these things you need to connect and remember. Questions are basically being framed merging the DNA repair mechanism with the mutations. So both of these topics, remembering both of these topics together and learning both of these topics together is important from the questions perspective. Then comes the transcription, where you need to study about both the prokaryotic and eukaryotic transcription mechanisms, in which the major 3 events you need to study. First is the initiation, second is the elongation, and third is the termination. In initiation also, what is abortive initiation? What is CTD phosphorylation in eukaryotes? What is its importance? CTD is basically the C-terminal domain phosphorylation and then the elongation, and in termination you need to study about the prokaryotic termination where row dependent and row independent mechanisms are involved. What are their differences? These differences you can learn through comparative tables. Yes. Here comes the use of the concept wheel. So for the transcription factors, you can make use of concept wheel to study this topic. The general transcription factors in eukaryotes with their roles, specifics of core promoters and regulatory sequences, activators and co-activators, all these things you can study with the help of concept wheel. Let me tell you how. So as we know that in eukaryotes, the transcription factors are involved and they come in sequence and perform specific roles. So what is the sequence of the events that are taking place? What kind of transcription factor comes at the starting, then at last? All these things you can learn through concept table. So here I've given an example how transcription factors comes in order and what all functions they perform. So as suggested, refer the concept wheels to remember the sequence and the names of the transcription factors. This can be quite handy for you, so make use of this. Then comes the role of promoters, activators and repressors. Part C, graphical based question and figure based questions are majorly being asked on the role of transcription factors, promoters and activators. Concepts could be merged with the recombinant DNA techniques, markers, mutations and operons and can be asked in exam. Such as I have taken an example here. This is a question which is being picked up from CSIR net exam. So here this question basically deals with the activity of the promoter. So for this slide also, I want you to pause the video, try to read the question what it is asking and try to solve yourself. If you get an answer, write it down in the comments. Then comes the post-transcriptional processing. Here you can make simple and crisp flowcharts for molecular events that are being involved during capping, polyadenylation and RNA splicing. Within RNA splicing also, you can make charts for alternative splicing and trans splicing. RNA editing is also important. Highlight the names of the enzymes that are involved in all these events. What are their roles? What are their functions? Direct questions sometimes are being asked in part C from all this concept. Then comes the RNA splicing and types. Here you can make use of concept tables to do the comparison between the self-splicing and spliceosome mediated splicing, group 1 introns and group 2 introns. What are the major types of enzymes that are involved? What are their roles? All these things you can do with the help of concept tables. How? Let me show you. So here I've taken an example where I am comparing group 1 introns with group 2 introns and spliceosomal splicing with self-splicing. So in this way you can make your own comparison tables and can study and revise these topics through these comparison tables. It will be quite effective for you and you can remember it for the time of exam. Then comes the diagrams. Diagrams are one of the very important things that you can make use of to learn or to understand the concepts of topics such as spliceosome mediated pathway. where you can take help of these simple diagrams that are being mentioned in your standard books, such as this diagram I have taken from the standard book Watson for molecular biology. So direct questions are being asked in part C from these concepts or these pathways. So here this diagram is simply explaining you what are the different type of events and which sequence it is going on for the spliceosome mediated pathway, where different types of S and RNPs come and play their role in removing the introns and converting the pre-mRNA to matured RNA. Yes. Then comes the genetic code. There is a huge table that is being given both in Watson as well in Leringer, but the good news is you don't have to remember or study each and every the code for each and every amino acid. The important codes that you need to remember are for differences between the universal and the mitochondrial codes, and what is the initiation code, AUG, universally used. What are the different types of termination codons, that is UAG, UGA and UAA. Basically, the numerical based questions are being asked from genetic codes in exam. One such example of numerical based question from genetic code topic is given here, where it is given that naturally there are 20 amino acids that are present in nature. But here we have assumed that 720 unnatural amino acids have been reported, and if all the 720 were to be accommodated in the genetic code, what is the minimum number of base that is being needed in a codon? So which answer should be right? For this, I want you to pause the video, try to solve this question and have an answer in your mind. Then I'll tell you what is the correct answer for this. So solution is here. As we know that the total number of different nucleotides that are present are majorly 4 types, that is A, T, C and G. So if we need to calculate the number of base in a codon, we can use the formula 4 to the power n, where n basically represents what would be the number of bases in the codon, and 4 to the power n, the value should be either greater or equal to 720. So for this, what we can do is the n should be 5, because 4 to the power 5 is equal to 1024. So in this way, you can solve these kind of numerical based question which comes in the topic genetic code. So answer B would be correct. I hope all of you would have marked the right answer. Then comes the antibiotics and their action. This is very important topic. Each year, one or two of these antibiotics are asked in exam. What are their mode of action and how and where they basically act upon during translation event, both in prokaryotes as well as eukaryotes. So with the help of table, you can know and memorize all these type of antibiotics and their roles and what are their targets. For the OPERON model, as I have told you earlier, lac operon and trip operon are very important. In lac operon also, you need to know about all the genes that are used in lac operon, that are involved in lac operon, what are their different roles and functions. And to understand the topic of OPERON model, you can make use of concept tables where you need to compare certain kind of terms and what are their concepts, such as what is positive regulation in OPERON, what is negative regulation in OPERON. Both of these things you can compare and make tables, concept tables. Another is inducible operon and repressible operon. This also you can differentiate and compare with the help of concept tables. This could be quite handy and you can memorize these things better. Yes, the experimental based questions are the favorite of CSIR net, which majorly comes in part C from the concept of operons. So here is a question that I have taken from one of the CSIR net paper, where it is given that this graph basically represents the expression of the tryptophan synthetase in E. coli cells in absence and presence of tryptophan, which is the substrate of trypoperon, in the medium. So if the two trip codons in the leader sequence of trypoperon is mutated to alanine, which of the following graphs will best represent the activity of this tryptophan synthetase in E. coli grown in the absence and presence of tryptophan? So out of these four options, which could be correct? If you have known the concept of trypoperon, that there are two types of regulatory mechanisms which are involved, then you can answer this question. So I'll give you some time to read the question and find the answer. Then I'll tell you what is the answer. Yes, for this, the option 2 is correct. Now we'll know how our option 2 is right option through this explanation. So trypoperon, as we know that there are two controls. First is the negative control, second is the attenuation control. In negative control, the that is tryptophan substrate, basically binds with the inactive repressor, inactive regulator protein, and it makes it active, which then goes and binds to the operator to stop the transcription of the operon. This is the first control mechanism that the tryptophan operon takes. And the second is the attenuation control, where it is being mediated by the two of the trip codons which are located in the leader sequence. So in the question, since there are two types of controls, when the trip concentration in the cell is very much high, the operon is turned off. When the trip codon is in the leader sequence is switched to alanine as given in the question, the attenuation control by trip cannot work anymore, because the whole of the codon, the two codons which are responsible for the attenuation control is being changed to alanine. Only the negative control of the operon will work at this time. Even in the presence of tryptophan now, some of the tryptophan will still be transcribed. Okay. Then amount of which will be lesser than the expression when trip is absent, but it will be more than the situation in presence of tryptophan, when both of these regulations are working. So this is the whole explanation of the question that we have solved here. So understanding these kind of concepts with the help of questions is very important, because you'll get an idea that in the exam, what are the different forms the exam asks the concepts on OPERON model. Then now I'll be sharing with you some of the quick tips which you can adopt during the revision of Unit 3. Firstly is make descriptive and colorful charts to revise the topics or to revise various factors that are involved in replication, transcription, translation, proteins which are the part of regulatory mechanisms and inhibitory mechanisms. All this you can revise with the help of charts, the colorful charts that you can make. You shouldn't miss the subtopics, because there are various questions which are being asked in combination with the statement-based questions that are being sourced from different subunits. So each of the subunit which I have mentioned here to study this unit is very important. You should learn all of these subunits. Missing them wouldn't be recommendable. Ensure to learn and master the recombinant DNA technology technique from unit 13, because most of the questions are being combined with this unit in the CSIR NET exam. Make sure you know or you remember the concepts related to mutations, which is majorly the part of unit 8. Practice the numerical type questions used in lab OPERON, as we have discussed, and the Meselson Stahl experiment also. I'm sure this session would have helped you, and you will adopt all of these techniques which you require to revise Unit 3 faster and ace in your CSIR NET exam. Thank you.